Hello everyone, this is Rohit Shankar Mane and welcome to my YouTube channel Rohit Academy. Today we are going to learn about my pharmaceutical microbiology laboratory. Now in this video lecture we are going to understand what is pharmaceutical microbiology, what is microbiology, what are microbiologists and what is pharmaceutical microbiology laboratory. Now what are instruments are used in pharmaceutical microbiology laboratory, which posts are available in the pharmaceutical microbiology laboratory we are going to study in this video. Now let's start the video. First we are going to understand what is microbiology. Now microbiology is the study of microscopic living organisms such as bacteria and fungi. The branches of microbiology can be classified into pure and applied sciences. Microbiology can be classified based on taxonomy. Pure microbiology encompasses numerous subdisciplines including mycology, bacteriology, and virology. Medical microbiology is perhaps the most well known because it deals with the role that microbes have in human illness. Other types of include environmental microbiology, food microbiology, and pharmaceutical microbiology, as these deal with the way microbes or microorganisms affect the environment, the food supply, and the healthcare industry. There is considerable overlap between the specific branches of microbiology with each other and with other disciplines, and certain aspects of these branches can extend beyond the traditional scope of microbiology. Now, what are microbiologists? A microbiologist is usually a person with a minimum of bachelor's degree in microbiology. Microbiologists usually work in hospital, clinical laboratories, food industry, environment research laboratories, and pharmaceutical industries. The role of the microbiologist is to investigate the growth and characteristics of microscopic organisms such as bacteria, algae, viruses, or fungi. Now, what is microbiology laboratory? Microbiology lab is the place where all microbiological tests and analysis occurs. It has special preparations in design and precautions as the hazard materials. Here is the microorganisms which might be infectious, which is different from chemical and physical lab, as here these pathogens can multiply or transfer out of the lab if precautions is not allowed. The design of the lab have to ensure that the floor, walls and benches in the lab does not support or permit microorganisms growth. The scientists working in the microbiology lab is always safe as long as they follow precautions and are focused in their work. Different equipment and instruments is used in microbiology laboratory. We will list some of the basic devices needed in microbiology laboratory in upcoming videos. Now first, posts in the lab. Now if you are working in quality control lab, then posts will be like, first you can start your career as a microbiologist trainee, microbiologist junior officer, microbiology senior officer, then microbiological junior executive officer, then senior executive officer, microbiological junior assistant manager, senior assistant manager, and last one microbiological manager. If you are working in the R&D lab of microbiology, then you can start your post from microbiology research trainee, microbiological research assistant, microbiological research associate, microbiological junior scientist, microbiological senior scientist, etc. Now we are going to understand some instruments from microbiological laboratory. Now first is the autoclave. We can see here autoclave diagram. Now autoclave mainly consists pressure gauge, pressure regulating device, where we can regulate our pressure in the autoclave. Then we have safety valve, lead handle, body handle, and here steam release valve and vacuum release valve. Now autoclave is a machine that uses steam under pressure to kill harmful bacteria, viruses, fungi, and spores on items that are placed inside a pressure vessel. The items are heated to an appropriate sterilization temperature for a given amount of time. First time autoclave was described by the Chamberlain scientist. It is used for sterilization of media, glassware, aprons, and different microbiological instruments at 121 degrees Celsius at 15 psi for 15 minutes, because at 121 degrees Celsius we can kill endospore forming bacteria. It is used in decontaminating biohazard waste also. Now next is the incubator. An incubator is a device used to grow and maintain microbiological cultures or cell cultures. The incubator maintains optimal temperature, humidity, and other conditions such as CO2 and oxygen content of the atmosphere inside. It is a device which is used to maintain growth of microorganisms. All types of microorganisms can grow in incubators such as sacrophilic, mesophilic, and thermophilic. Next one, laminar airflow. Now it is used to create dust-free environment. It contains HEPA filter. Laminar airflow is used to separate volumes of air or prevent airborne contaminants from entering an area. Laminar airflow hoods are used to extract contaminants from sensitive processes in science, electronics, and medicine. Next is oven. Laboratory ovens are ovens for high force volume thermal convection applications. These ovens generally provide uniform temperatures throughout. Process applications for laboratory ovens can be for annealing, dye bond curing, drying, polymer baking, sterilizing, and other industrial laboratory functions. What dry heat sterilization does heat inflicts damage by oxidizing molecules leading to the organism's death. Resistant spores are killed by exposing them at higher temperature for a long period of time in oven. Next, pH meter. The device is used in the laboratories that measures the hydrogen ion concentration in water-based solutions to determine the acidity or alkalinity of the solution. It is used to measure the pH of soil in agriculture area, then water in the from water microbiology, swimming pools, environmental remediation, and etc. Loop to Nichrome wire loop. We can say that only Nichrome wire loop. A nichrome wire loop consists of looped nichrome wire which is fixed into a brass handle. It is easy to sterilize and reuse because nichrome wire resists deterioration with repeated heat or cooling cycles. Plain sterilization is very simple method for killing microorganisms on an nichrome wire loop. This wire loop is used to transfer microbial cultures from one culture to another culture. Bunsen burner. A Bunsen burner is also used in microbiology laboratory to sterilize piece of equipment and to produce an update from that forces airborne contaminants away from the working area. Three types of flames are obvious when adjusting the air and gas mixture. Yellow flame, safety flame, is the coolest flame at 300 degrees Celsius. Blue flame can reach temperatures approximately 500 degrees Celsius and most invisible in a bright room. Microscopes. Now, microscope is the important instrument in the microbiology laboratory. It is used to identify and visualize the microorganisms. Power objective lenses with high resolution as well as observation techniques such as phase contrast microscope, dark field microscope are the key observations of some microorganisms. Cotton. Now, it is a white, clean, and soft non-absorbent cotton used for plugging test tubes and other glasswares. And non and absorbent cotton sinks the water, but non-absorbent cotton floats on the water because it's a fatty substances. And absorbent cotton, because of its softness, is widely used in surgical dressings, cosmetic purpose, etc. Whereas non-absorbent cotton is widely used in plugging of test tubes and glasswares, especially used in microbiology laboratory. If you have any question, you can comment or you can mail me. Thanks a lot.